All right, let's continue talking about the second line of defense. So, in the last video, I talked about inflammation. So now we're going to talk specifically about the phagocytosis part of inflammation. So, phagocytosis is done by several different types of cells. So, neutrophils and macrophages mainly, right? Remember that monocytes is just the name for a macrophage when they're in the bloodstream. So it's really like the neutrophils and the macrophages. They move to the site where the infection is. They identify anything that doesn't have a self marker, right? So remember anything that's foreign to you, and they are going to grab onto it, engulf it into a vesicle called a phagosome, fuse that with a lysosome, and they're going to destroy that foreign material. They're also responsible for extracting little bits of those dangerous pathogens and creating what is, I don't want to say creating, but pulling out what is called antigen that's going to cause the third line of defense or stimulate the third line of defense, so we can have some memory of that interaction for later. But that part of phagocytosis is, part of the third line of defense, which will be discussed in the next series of videos. All right, so this just kind of shows a picture, right? In your bloodstream, they're called monocytes, and then once they're in the tissue sites, they're called macrophages. The specialized macrophages that you see when we look at A&P slides, which I mentioned in a previous video that you might be familiar with, are those alveolar macrophages, right? Those are macrophages that are found in your lung tissue. They're there all the time. That's where they hang out. Why? Because that's a place that's exposed to the outside world and, experiences a lot of pathogens, plus very important to protect. When you look at liver slides, and study liver tissue, you see Kupffer cells. What are Kupffer cells? They're just specialized macrophages that are found in the liver cells that are there to help prevent infection. Langerhans cells, this is in your skin, right? Again, your skin is quite often exposed to the outside world. These are macrophages that are hanging around in that tissue space, right? Just waiting to engulf anything foreign that comes around, right? So once the macrophage has been recognized something that is non-U, right? Doesn't have a self marker, doesn't have that major histocompatibility complex. It will grab onto it and it will engulf it or phagocytize it into a vesicle, right? And you can see it's wrapping around these invading organisms, right? And the vesicle that it forms is called a phagosome. The lysosomes, remember, have those reactive oxygen species. So here you can see there's superoxide anion, there's hydrogen peroxide. And when the phagosome fuses with the lysosome, what do we call it? We call it the phagolysosome. And the phagolysosome, all of that reactive oxygen starts to break down that bacteria, kill the bacteria, right? And then we can release that debris. That debris could be antigen, right? It could be part of the invading pathogen that would then go off and stimulate an immune response in the T cells and the B cells, right? That debris is going to drain through the lymphatic vessels, be filtered out through the spleen, somehow being cleared out from the site of the infection. All right, so that's phagocytosis and how it sort of ties into the bigger overall picture of the immune system. The third part of the second line of defense, right? So we have inflammation, phagocytosis, is interferon. Interferon is a cytokine, a specialized cytokine that is going to bind to cells and change their gene expression. Which, if you just think about that, it's a compound that we have that can bind to a cell and change its gene expression. There's a lot of hope for how that could be harnessed as a drug. So if we think about cells that have been invaded by a virus, right? What's happening inside of that cell? The virus is replicating and taking over the host machinery and doing all of this stuff in order to create more virons that are then going to be released out into the body or out into the world. So what does interferon do? Interferon can bind to that invaded host cell and change the gene expression. It can turn it off. It can tell it to, stop replicating DNA, right? Because it knows that it's infected by a virus. And we'll look at some pictures here in a minute that kind of demonstrate that. It also can be a tumor suppressant, right? So cancer cells are cells that are sort of growing out of control. There's some sort of um, problem with the gene expression of those cells, right? So interferon can suppress tumors. Right. So this sort of demonstrates how two cells adjacent to each other could be interacting through the formation of interferon, right? So here we have a cell, it's been invaded by a virus, it's starting to replicate, it recognizes that there's something wrong here, right? So what does it do? Is it produces interferon that it pumps out of the cell. That interferon is a cytokine, a chemical signaler to this neighboring cell. It binds to a surface cell receptor and says, hey, stop making proteins, block, replication. So stop transcription translation, right? So it's a chemical signaler between cells, right, to prevent or to change the gene expression within another cell. All right, the last thing that I'm going to talk about then is something called the complement system, right? So the complement system is a very complicated network of proteins, like this cascade of different proteins that come together to form a membrane attack complex. I always call it a protein donut. You're essentially building a protein donut either in the surface of a virus or certain types of bacteria, right? And if you build a protein donut or a membrane attack complex in the surface of these things, it essentially creates a hole or a pore that will lead to the lysis of that. It can start in different ways. The classical pathway for the complement system is for it to be triggered by an antibody. So it's correlated with the third line of defense. There's a lot of overlap and interaction between all of these players, right? So in the next series of videos, I talk about where do antibodies come from, what is their function. So just remember that one of the functions of an antibody is to help to trigger this complement system. Now, the complement system, though, is still considered to be a non-specific host defense because it just recognizes that there's antibody there that your body made. It's not recognizing anything specific about a foreign pathogen that's come in and responding to it. It's just like, oh, there's your antibody here. I'm going to build protein donuts. Right? So the classical pathway, you can see all of these series of different proteins. Each one of these different things is a protein. And when I took immunology, I had to memorize all of these steps, like the C1 complex binds to the antibody, which triggers the C4 and the C3b and the C2b to come together, and then they make the C5. It's just a lot of building and breaking and forming of what essentially becomes a protein donut, your membrane attack complex. It's a ring of protein with a hole in the center. And here's a picture you can see of an organism that has had a whole bunch of protein donuts or membrane attack complexes built into its surface. So it's essentially just creating holes, big holes all over that are going to, eventually lead to the lysis of that cell. So that's what the complement system is. So just remember, just to kind of do a quick overview, the 4 parts of the second line of defense of the immune system are 1, inflammation, which is bringing in immune cells and fluids to the site of an infection so that they can be trapped, destroyed, and the area cleaned up, right? The second part, which is tied into inflammation of the second line of defense, is phagocytosis. So macrophages, neutrophils, engulfing and destroying the foreign material or the foreign invading pathogen. The third is interferon, specific to more often viruses, right? It's a cytokine that chemically signals between adjacent cells and can change the gene expression so we can suppress tumors or we can prevent viral replication within host cells. And then lastly is this complement system, which is triggered by antibodies. It can also happen in other ways without antibodies present, but is essentially a non-specific formation of a membrane attack complex, a protein donut that drives or inserts a bunch of pores into an invading organism that will then cause that cell to lyse. All right, so that's non-specific immunology. And in the next week, we'll go into the specific part of immunology where we're responding to something specific about the invading foreign organism, as well as how we remember it later so that our immune system is more efficient in fighting infection in the future.